What is culture media? Culture is growing the organism. Culture is to grow the organism. Culture is media on which we grow. Usually we put it on petri dish. Why it is called petri dish? Because it was discovered by Petri. It's named after him. It's a petri dish. So for first instances, first of all, we should know why do we grow the organism. Number one, we grow the organism, isolation for diagnosis of infection. We grow the organism in the laboratory for isolation of organism. For example, if it is an enteric fever, we have to grow whether it's S. typhi, paratyphi A, B, C. We have to grow in the media, culture media. If it is sore throat, we have to grow streptococci. If it is diphtheria, we have to grow the diphtheria organism in the culture. So isolate the organism which is responsible for the disease. To find out the organism which have infected the organ. Okay, whatever infection, osteomyelitis, whatever it is. First of all, you have to isolate the organism to find out the what organism has caused the infection. That's why you have to grow the organism first and foremost. To find out the infection, find out the diagnosis, the diagnostic modality. We have to find out why it has caused, which organism has. Next is to do the antibiogram. Antibiogram, we call it as culture and sensitivity. We grow the organism and put various antibiotic discs to find out whether the organism is resistant or sensitive to the particular, for example, ciprofloxacin, nitrofurantoin, uh, ceftriaxone, gentamicin, meropenem, imipenem, all those things. We put the antibiotic disc and look for the zone of inhibition. Depending on the Kibri-Bar chart, we decide whether the organism is sensitive or resistant to infection. Organism is sensitive or resistant. So all the bacteria which are grown, are antigens. All the bacteria which are grown in the culture are antigens. So antigen is defined as when you inject into the body, it should be able to provoke either cell mediated or antibody mediated immune response. It should be foreign to the body. So whatever organism we are growing is used as an antigen, and it is used for antigen antibody reaction. These are called as serological tests. These are called serological tests. Antigen antibody reaction we do for the diagnosis of infection. For the diagnosis of for the diagnosis of infection. For the diagnosis of infection. For the diagnosis of infection. Now, antigen what you have grown in the laboratory, bacteria which are grown in the laboratory, which can be prepared what is called as vaccine. Vaccine. You know the story of vaccine, isn't it? Anybody tell me what is a vaccine? Story of vaccine. Way back there was a person called Edward Jenner. He suffered from the smallpox infection at the age of 8. He underwent variolation. He salvaged, and he's a doctor. In India, we used to practice variolation. We used to take the healing scab make it in a powder, insufflate into nose, or make a injection form. And sometimes live virus used to be there. People used to die of this smallpox infection. Moved to the London where they practice variolation as a OT procedure. And once Jenner was told by Sarah Mims, milkmaid, that she has suffered from cowpox and she will not suffer from smallpox. Cow in Latin means vakka. Cow in Latin means vakka. Cowpox virus is used to prevent smallpox. Cowpox virus was used to prevent smallpox. So all prophylactically given antigens are called vaccines. All prophylactically given vaccines are called vaccines. So we used cowpox virus, which is antigenically similar to smallpox, and which gives a milder disease, and people will survive from it. And today there is no smallpox. It's called globally eradicated. In 84, 85, last case was reported from Bangladesh. Now there is no smallpox. Okay. So next is to find out the source of infection. To find out the source of infection. For example, all of you are down with diarrhea. You look for the source of infection in your water, where you get from water, where you drink water from. Water is checked whether it contains any infection. Now, next thing is very important. Sometimes I heard of story of Mary Mellon. She was a cook. She was nicknamed as Typhoid Marian. And everybody with me, carriers are defined as those who carry the microorganism without suffering from it. Patient is a carrier. Patient who is recovering from infection is a carrier. He's called convalescent carrier. Temporary carrier, carrying microorganism less than 3 months. And permanent carrier, carrying for more than 6 months. And there is paradoxical carrier, carrier acquiring from another carrier. Carrier from a carrier can be asked as a 5 marks question. So now sixth one is to identify the carrier. To identify the carriers. Sixth one. So once again I'll repeat. First of all, we isolate the organism for diagnosis. Everybody. We isolate the organism in the laboratory for a diagnosis of disease. To find out whether the organism resistant or sensitive to particular antibiotic. This report is called as antibiogram, and this is all procedure is called culture and sensitivity, growing and finding out the antibiotic resistant or sensitive to particular antibiotic. The third one, the antigen which you have grown, used for a serological test. For example, I said Brucella test, Brucella agglutination test, Vidal test, Wheel Felix, Paul Bennett. So many serological tests are there. These antigen can be attenuated by heat killing, and phenol preservation. We prepare vaccine. There are at least half a dozen of vaccine available current in the market to prepare the vaccine. Next is to find out the source of infection. Some dozen of people get diarrhea in the hostel. The source we look for the drinking water. Next is to identify the carriers. Whether it is she's carrying in the feces, she's carrying in the urine or something like that. So it's very important. Now, classification of culture media is on the consistency. Consistency, it is classified as solid, liquid, and semi-solid. It is classified into solid, liquid, solid, liquid, and semi-solid. It is classified into solid, liquid, and semi-solid. So most of the students, when I ask in examination, in that tension, they said solid, liquid, and gas. There is no gas media. Okay. There is anaerobic media, not gas media. Okay. Don't answer solid, liquid, and gas. Three states: solid, liquid, and semi-solid. Okay. Then other, depending on the use, they are classified into simple media. Repeat with me: simple media, enriched media, enrichment media, simple media, enriched media, enrichment media, differential media, selective media, selective media, media for biochemical reaction, media for biochemical reaction, media for anaerobic cultivation, anaerobic organism cultivation, media for anaerobic organism cultivation, anaerobic organism cultivation. Repeat with me: medias are classified depending on the use: simple media, enriched media, enrichment media, differential media, selective media, media for biochemical reaction, and media for cultivation of anaerobic organism. Why it is important to study culture media is to pass your examination. One. Number two, it is given as a spotter, 1 mark spotter in the examination. It is given in a your applied exercise. Media is kept. You have to identify. Third thing, it will come as short note, or sometimes culture media can be asked as a full question, as a 10 marks question. So that's why we are teaching you. Otherwise, we have no pleasure in this thing. Because you may think, what is this? Some colorful glasses, colorful biochemical reaction. This is all you read, understand. Once you pass, only if you take MD microbiology after that, then we'll teach you intensively what is the content and all those things. At least for passing an Viva examination and writing in your theory examination. And this is the basics. Because one of the postulates is you should isolate the organism in pure culture. That's one of the postulates of Robert Koch. Okay, we'll continue in the next class. Thank you very much. So whatever any number, I don't mind. Even if two student, one student comes also, I'll take it. This is a like tuition. Okay. Yeah. If less students are, it's better.